What's up, guys? How are you all? I hope you're super excited for this series where we are going to complete NCERT reading of class 11 and 12 biology as well as chemistry. Please do comment down below if you want me to do physics as well. As of now, our first target is class 11 biology. So, invite your friends, like, share, and subscribe. And now let's not waste any more time and get started with the chapter. Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over a million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, or reproductive systems. These features are used as the basis of animal classification, and some of them are described here. Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, these cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor activities occur among these cells. In coelentrates, the arrangement of cells is more complex. Here, the cells performing the same function are arranged into tissues, hence is called tissue level of organization. A still higher level of organization, that is organ level, is exhibited by members of Platyhelminthes and other higher phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms, and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. Open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and closed type, in which the blood is circulated through a series of vessels of varying diameters, arteries, veins, and capillaries. Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, that is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organisms into two identical halves, it is called radial symmetry. Coelentrates, ctenophores, and echinoderms have this kind of body plan. Figure 4.1a. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Figure 4.1b. Diploblastic and triploblastic organization. Animals in which the cells are arranged into two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. Example, coelentrates. An undifferentiated layer, mesoglia, is present in between ectoderm and the endoderm. Figure 4.2a. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and the endoderm are called triploblastic animals. Platyhelminthes, two chordates. Figure 4.2b. Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity which is lined by mesoderm is called coelom. Animals possessing coelom are called coelomates. Example: annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. Figure 4.3a. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates. Example: Achyhelminthes. Figure 4.3b. The animals in which the body cavity is absent are called acoelomates. Example: Platyhelminthes. Figure 4.3c. Figure 4.1a. Radial symmetry. Figure 4.1b. Bilateral symmetry. Let's look at figure 4.2 showing germinal layers: A. Diploblastic, B. Triploblastic. Let's look at figure 4.3. Diagrammatic sectional view of A. Coelomate, B. Pseudocoelomate, C. Acoelomate. Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomena is known as metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates. Example: Porifera to echinoderms. Classification of animals. The broad classification of animals based on common fundamental features as mentioned in the preceding sections is given in figure 4.4. Figure 4.4. Broad classification of kingdom Animalia based on common fundamental features. Kingdom Animalia, multicellular. Levels of organization. Cellular level. Symmetry, mostly asymmetrical. Body cavity or coelom, acoelometer. Phylum Porifera. Level of organization, tissue level. Symmetry, radial. Body cavity or coelom, acoelometer. Phylum Coelentrata or Cnidaria and Ctenophora. Level of organization, organ level. Symmetry, bilateral. Body cavity or coelom without body cavity, that is acoelomates. Phylum Platyhelminthes. Level of organization, organ system. Symmetry, bilateral. Body cavity or coelom with false coelom, that is pseudocoelomates. Phylum Achyhelminthes with true coelom, that is coelomates. Phylum Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. Echinodermata exhibit radial or bilateral symmetry depending on the stage. The important characteristic features of the different phyla are described. Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. Figure 4.5. These are primitive, multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores, ostia, in the body wall into a central cavity, spongosoil, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Coenocytes or collar cells line the spongosoil and the canals. Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate. Hermaphrodite, that is, eggs and sperms, are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Figure 4.5. Examples of Porifera. A. Cycin. B. Euspongia. C. Spongilla. Examples: Cycin, Scypha, Spongilla, freshwater sponge, and Euspongia, bath sponge. Phylum Coelentrata, Cnidaria. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. Figure 4.6. The name Cnidaria is derived from the cnidoblasts, so cnidocytes, which contain the stinging capsules or nematocysts present on the tentacles and the body. Cnidoblasts are used for anchorage, defense, and for the capture of prey. Figure 4.7. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening mouth on hypostome. Digestion is extracellular and intracellular. Some of these cnidarians, example corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. Figure 4.6. The former is a sessile and cylindrical form like Hydra, Adamsia, etc., whereas the latter is umbrella shaped and free swimming like Aurelia or jellyfish. Those cnidarians which exist in both forms exhibit alternation of generation, metagenesis. That is, polyps produce medusae asexually and medusae produce the polyps sexually. Example, Obelia. Examples: Phycelia, Portuguese man of war; Adamsia, sea anemone; Penicula, sea pen; Gorgonia, sea fan; and Meandrina, brain coral. Let's look at figure 4.6. Examples of coelentrata indicating outline of their body form. A. Aurelia, Medusa. B. Adamsia, polyp. Let's also have a look at figure 4.7, diagrammatic view of cnidoblast. Phylum Ctenophora. Ctenophores, commonly known as sea walnuts or comb jellies, are exclusively marine, radially symmetrical diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated comb plates which help in locomotion. Figure 4.8. Digestion is both extracellular and intracellular. Bioluminescence, the property of a living organism to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Examples: Pleubrachia and Ctenoplana. Figure 4.8. Examples of Ctenophora. Pleubrachia. Awesome guys, so this marks the end of the first section of the chapter. I hope you liked the video and do not forget to watch the next part of the chapter. You will find its link in the description box. Please share your thoughts in the comment section and don't forget to like, share and subscribe. Let's meet next Friday to continue the series with a new chapter. Bye.